Okay, so this is part 3 of his The Cell Really a Machine by Daniel J. Nicholson. And I believe we left off on page what? Right here?
Wherever section 5 is. I'm not sure what page that was.
Yep. Okay, there's table 3, and then we read this part. Now we're down at section 5.
There we go.
So here is where we are going to start, boys and girls. Okay.
I'll start.
Go ahead.
Cellular behavior, deterministic or probabilistic? So far, I have shown how the MCC, which is the machine concept of the cell, has provided the theoretical foundation for our traditional understanding of the cell's internal architecture, of the macromolecular complexes that compose it, and of the transport processes that take place within it. But its influence does not end there. The MCC has also shaped the way we think about how the cell behaves, as I will argue in this section. What a cell does is largely determined by its internal makeup. This makeup is constituted by a complex network of metabolic, regulatory, and signaling pathways that, as I have already discussed, are often misleadingly conceptualized in analogy with electronic circuit boards. These pathways, when prompted by internal or external cues, generate specific behavioral outputs that allow the cell to perform its various functions and respond to changes in its environment. As these pathways have a genetic basis, the behavioral outputs they specify are themselves dependent on how and when the pertinent genes become activated and transcribed in the cell. Consequently, in order to understand the basis of cellular behavior, it is necessary to consider how the all-important process of gene expression is initiated.
Okay, and we're going to go down here. Okay.
Basically, an intracellular or extracellular signal, an inducer, triggers a cascade of biochemical reactions that causes proteins called activators, which are a class of transcription factor, to bind to specific sites in the DNA known as enhancers. Upon binding, the activators interact with other proteins that recruit RNA polymerase and its associated transcription factors, collectively referred to as the pre-initiation complex, to the promoter region of the target gene, where it begins the process of transcription. Thousands of transcription factors have been identified in the past few decades, as have the enhancer and promoter regions of countless genes. But despite this wealth of information, there has been considerable debate regarding the precise way in which transcription is regulated and modulated. It has long been known from numerous experimental studies that when cells are treated with varying intensities of an inducer and the gene product mRNA or protein corresponding to a specific gene is assayed, the level of gene product changes in a smooth, dose-dependent manner. Specifically, a gradual increase in the concentration of the inducer usually results in a proportional increase in the expression of the gene. In order to make sense of this observation, it was generally assumed that cells adjust the rate of expression of a responsive gene progressively and linearly from zero to its maximum output in direct proportion to a rising concentration of an inducer. This came to be known as the graded model of gene expression. It is sometimes alternatively referred to as the rate, analog, or rheostat model, and it remained the dominant view of gene expression until the end of the last century. It is a model that clearly exemplifies the MCC, as it construes the modulation of gene expression as a continuous, linear, mechanical process akin to depressing the accelerator on a car. It is also a thoroughly deterministic model, as it suggests that gene expression, and by its implication, most cellular behavior, can be anticipated, computed, and predicted in principle, at least from the knowledge of the initial conditions, such as the concentration of the inducer. It therefore fits well with the classic mechanicist idea that the cell is endowed with a genetic program analogous to a computer program, which controls and reliably executes its operations in a predetermined way. When considering this model, it is important to bear in mind that until recently, gene expression and cellular behavior, more generally, could only be studied by looking at large populations of cells. If one uses conventional molecular biology techniques such as northern blots, microarrays, and reverse transcriptase polymerase chain reaction, did I get that right?
Yep, that's
Yeah.
the
Okay,
PCR.
that's just a bit much for me.
When they
The
talk
only
about
way to gather, I'm sorry, what?
when they talk about PCR, that's what they mean.
Okay,
Polymers, they amplify it,
cool.
basically duplicate the hell out of it, and then they can figure out whether it was there. That's what the ENCODE project is one of the things that they're using. They can take like one or two molecules and amplify them to the point where they can do an assay on them and figure out that they had been transcribed. So it allows you to find really, really small concentrations, I believe.
Yeah,
If I got
and that's the thing about PCR as well, is that it's exceptionally sensitive, so it doesn't need very much to make a lot.
right. Okay.
Cool. So if one uses these methods, the only way to gather together enough gene product to reach a detectable threshold is to grind up vast numbers of isogenic or genetically identical cells grown under the same conditions and then measure the amounts of the relevant mRNA or protein in the homogenate. What this means is that although the goal is to understand the behavior of an individual cell, one proceeds by studying the behavior of a population of cells. The consequence of doing so is that the specific behavioral patterns of individual cells are averaged out across the entire population, and then this can mask differences between members of the population. In the past, this methodological limitation was not considered a problem because molecular biologists habitually assume uniformity of the cell populations that serve as starting material for experimental analysis. If all cells are presumed to be identical, and one is therefore dealing with a homogeneous population, then one can confidently infer that the average behavior of the population as a whole accurately reflects the individual behavior of each cell in that population. Notice that this assumption, like so much else I have discussed in this paper, is grounded in the MCC. Just as cars manufactured in an assembly line according to the same model design will behave almost identically, cells endowed with the same genetic program and grown in the same conditions are expected to behave almost identically. And I yield the floor to another one of my colleagues.
That would be immutable.
Let's see.
The
The
situation.
the situation has changed radically in recent years. Technological advances in experimental use of fluorescent reporter proteins and the development of new microscopy techniques have granted us unprecedented access to the real-time dynamics of individual molecules and single cells. As we have begun to monitor biological processes on a cell-by-cell and molecule-by-molecule basis, it has become apparent that there exists a hidden world between population averages. Being able to precisely measure the distribution of cellular behaviors across a population, as opposed to merely relying on the average behavior of the whole population, has unexpectedly revealed that even isogenic cells subject to the same environmental conditions behave quite differently from one another. There is no such thing as a perfectly homogeneous population of cells. All cell populations exhibit some degree of heterogeneity. Gene expression offers perhaps the clearest illustration of this heterogeneity. Single cell studies strongly suggest that increasing the concentration of an inducer in an isogenic population does not lead to a gradual increase in the rate of transcription in every cell in the population, as hypothesized by the graded model, but rather results in the recruitment of a rising number of cells that respond in an all or nothing fashion once their particular activation thresholds have been reached. In other words, in each cell of the population, the target gene is either maximally expressed or it is not expressed at all, and the probability of its expression in every cell rises as the concentration of the inducer increases. In addition, once a cell begins to express the gene, the rate of its expression remains largely unaffected by further increases in the concentration of the inducer. with regards to each of the genes it contains, a cell appears to exist in one of two metastable functional states. It is either on or off. This is generally known as the stochastic model of gene expression. It is also referred to as the binary, digital, or threshold model, and has become very widely accepted in recent years. In direct contrast to the deterministic character of the graded model, the stochastic model is inherently probabilistic. Each cell in the population exhibits a specific and distinct probability to respond to a given concentration of inducer, and this probability can vary wildly, even among members of the same isogenic population. According to the stochastic model, the regulation of gene expression is accomplished by modifying the probability that the pre-initiation complex will come together successfully and bind to the promoter of the target gene. Activators and enhancers are presumed to act by increasing the likelihood that the promoter will be transcriptionally active at a given moment, but do not affect the rate of mRNA production once transcription has begun. The random switching of the transcriptional apparatus between active and inactive states is deemed to generate short and sharp bursts or pulses of transcriptional activity, which result in corresponding bursts of translational activity. The hypothesis that proteins are synthesized in bursts has been subsequently verified by single molecule experiments. Bursting behavior in gene expression has been reported not only in bacteria, but also in yeast, mammalian cells, and developing embryos. Even different nuclei in a single multinucleated syncytium, such as a muscle fiber,
Syncytium.
syncytium, such as a muscle fiber, which share not only a common environment but also a common cytoplasm, have been found to display disparate bursts of transcriptional activity.
Okay. Syncytium means that one cell with multiple nuclei, and like in the development of the Drosophila, that becomes very important. So we have a illustration here. I wonder, should we look at that first, or
Sure.
should we wait until it's talked about?
Let's read the next paragraph and then jump back up and read this.
You get there? Okay. Gotcha.
With all this evidence in support of the stochastic model, it may seem difficult to understand why the graded model remained the orthodox view for as long as it did. But the answer is quite straightforward. When the only way to measure gene expression in a cell was to assay the total amount of mRNA or protein produced by an entire population of cells, it was simply not possible to discriminate between the two competing models. Indeed, the aforementioned observation that progressively raising the concentration of an inducer results in a proportional increase in the expression of the corresponding gene is perfectly consistent with both models, as Figure 5 shows. The level of gene expression in the population as a whole could reflect similar levels of gene expression in all cells, as postulated by the graded model, or the statistical mean of different subsets of cells, either expressing or not expressing the gene, as postulated by the stochastic model. It was not until single molecule methods were developed that it became possible to examine gene expression on a cell-by-cell basis, and thereby distinguish the two models experimentally. The study of gene expression illustrates rather dramatically how being forced to average out data across a biological population due to methodological limitations can lead to critical losses of information pertaining to the phenomenon under investigation.
Okay. So our illustration up here, let's see, I make that a little bit bigger. No, I can't. Okay.
Yeah, okay.
Well, I could make it bigger like this.
Here we go.
Can you read it? Okay.
Yeah. So the text where it says graded versus stochastic models of gene expression, raising the concentration of an inducer in a proportional gene and the expression of the relevant gene in an isogenic population of cells. If gene expression is measured by assaying the total amount of mRNA or protein produced by the population, it is not possible to distinguish between stochastic and graded transcriptional responses, as both are consistent with the population level observations. Single molecule methods, however, have recently enabled gene expression to be studied on a cell-by-cell basis, and this has revealed that most of the cells exhibit an all-or-nothing stochastic expression pattern. A figure adapted from Craigstein, 1998 National Academy of Sciences.
Yeah.
So what it's showing is that in the graded response, um, the gray represents the amount of inducer, and as you move from left to right, the more you increase in concentration of the inducer, which the more dark is the greater concentration, the more the response the cell gives.
Yeah, and
How,
it's interesting, but if you look at the columns and think about this for a while, we're missing some things in the graded response, basically. Whereas in the stochastic response, you can see what's going on. In like a development of the embryo, you'll find that there are like the first column with nothing on will be one, it'll be one particular developmental pathway, and then the next two with a certain percentage will be another one, and then the next two will be still another. So you have basically three different switches rather than just this graded thing, right? So you can see that you can end up with what they call the French flag model of development, where you have red, white, and what is the other one in the French flag?
blue.
Is it blue? Okay, so you can have like a red, white, and blue response, or even more finely graded than that based on what's going on. And and then it becomes very complicated because they enter into other pathways. So it's kind of important that we understand that. It's a important bit about the development of the in the embryo. So
Well, remember, right? This is just measuring, like in the case of the stochastic response, right? It's measuring that for one particular inducer over time, right?
yep.
Or the for well, one, yeah. So imagine, right? With cells, you have thousands of genes being expressed and thousands of inducers for those genes, right? So I mean, this view, I mean, is the one dimensional idea gets it across, but technically, right, it's not as if though a cell is just transcribing one gene at a time, right? It's doing many other things,
Yep.
many things to take care of and to maintain. And so, in fact, this plot is going to be very multidimensional if you're studying the behavior of a cell and what molecules it's expressing and what concentration and how that relates to the concentration of inducer.
Yeah, there are some details that we miss, basically, that are many more pathways are possible with the stochastic model
Now,
than
do
with
you
the
consider
graded
that
model.
a problem with the paradigm or just a problem with available methodologies? Were both methodologies of looking at the cell available throughout time, or did we just develop some new technology in the 90s and we're able to see that not each cell receives a proportional amount of the inducer?
Yeah,
I
that would
need
have.
to look that up because I'm tempted to say that
It's both.
well, sort of. I mean, let's see.
We discovered that this situation existed as we got more. Uh, Hang on a second. As we got more sophisticated methodologies, but still, the paradigm would have kept us looking at averages or taking large amounts of the same kind of cell and grinding them up and doing it that way. But with the paradigm shift, it allows us to look more carefully into what is really going on. So it's a little of both. So the
So with
paradigm.
the new paradigm, you're more motivated to look at things on an individualistic basis rather than an average basis.
What was the question? Sorry, I must have missed
So
that.
I just said, so based on the paradigm you use, you're more likely to look at things on a more individual cell basis rather than a average population type basis.
Yeah, I mean, keep in mind, right, that this is true, like with all experiments, that whenever you have a sample, like there's going to be some variance within that data that you obtain. And part of the issue too is like when you take large samples, as like this paper's pointing out, if you take large samples of cells, right, it's quite possible that there's outliers in terms of how much of something is being expressed or whatever particular thing they're trying to study, right? But those outliers kind of get subsumed by the fact that that the data set is just very, very large, right? You might be studying thousands of cells in a population. So, as these numbers are, getting large, outliers don't really become so apparent. At least if you're doing kind of like a graded response, if you're basing the behavior based upon an average population. Whereas if you're doing it on an individual basis, you can kind of see where those where those murky details are hiding, right? You can see almost, that, oh, well, that's interesting. It seems that some of these are expressing this gene in a very non-uniform way, or they're actually doing it in a in a type of way that we wouldn't expect, right? So it's not just a random error. It's not just a machine, messing up and giving you wrong numbers. No, there's actually something further here, right? There are like in the stochastic model, there are members of the population, in fact, that may not be expressing it at all, but some that might be expressing it, full speed ahead.
Yep. And there's also the possibility for, when you consider neutral theory, that there are a lot of mutations. There are a lot of pathways that are not actually doing anything or connecting to anything, but they're partial pathways. They haven't joined up with anything yet. So what this tells us is that there are many hidden pathways that are probably churning around, waiting to hook up in some significant or useful manner, or in some incredibly bad, cancerous manner. So this one of the reasons why they're looking into this is that some of those pathways can lead to the death of the organism, right? And others can lead to the evolution of the organism. And this just gives us more statistical probability that a new pathway could evolve by looking at this. And that's something I've been harping about for quite a while. So anyway. Okay, moving on.
Yep.
My turn?
I guess so.
I read badly, so I won't read too much because I'm a high school dropout. Intriguingly, it has been suggested that the distinction between graded and stochastic models of gene expression is conceptually similar to the difference between Newtonian and quantum mechanics, and it offers similar intellectual challenges. This theoretical comparison is not as far-fetched as it may seem. The stochastic model regards cellular response patterns as state transitions, which are reminiscent of thermal or quantum phase transitions. Moreover, the precise timing and frequency of the switching between active and inactive transcriptional states is impossible to predict, as it is not a mechanical process, but a stochastic one. In the absence of deterministic certainties, genes in a population cannot be considered to be either active or inactive at any given instant. Instead, they can only be attributed a probability of being active in a particular cell at a particular time, even if this probability can sometimes be very close to 0 or 1. And that kind of flies in the face of the idea of calling something functional or not functional. There are so many spaces in between that we have to consider between functional and not functional. So it doesn't make any sense to even talk about junk DNA or not junk DNA. As soon as you say the phrase junk DNA, you're not even wrong. Okay. The variable flickering of transcriptional activity in different cells is one of the major causes of heterogeneity in isogenic populations. But where exactly does this cell-to-cell variability in transcriptional activity come from? The answer becomes apparent when we remember that gene expression is a molecular process, and like all molecular processes, it is inherently stochastic. Given that it takes place in an environment that is subject to the chaotic dynamics of Brownian motion, each step in the process relies on fortuitous encounters between molecules that are randomly moving about as a consequence of thermal agitation. Evidently, these molecules must be at the right place and at the right time, not to mention in the right vibrational state, for them to be able to participate in the appropriate reactions. The unpredictability of the whole process is further amplified by the fact that the participating molecules in each step are present in the cell in very low copy numbers, as this decreases the chance of successful interactions between them. DNA is the most extreme example, since there are usually only one or two copies in a cell at a given time, but mRNA and most regulatory proteins and enzymes are present in remarkably small numbers as well. Other contributing factors to cell variability in gene expression include variations in the topological configuration of the nuclear architecture. And that is that histone binding to the lamin inner surface of the nucleus,
Yep.
chromosome regions, and the uneven partitioning of the cytoplasmic contents during cell division. Of course, gene expression is only one of the many cellular processes that, due to the inherent stochasticity of the molecular interactions that underpin them, generate heterogeneous responses in isogenic populations. Okay. I'm done.
All right.
Scotty.
I will now begin to read. From a theoretical perspective, the discovery of non-genetic heterogeneity in isogenic cell populations came as a huge surprise. At first, molecular biologists struggled to make sense of it, as it is a difficult phenomenon to accommodate within the mechanicist interpretive framework of the MCC. After all, as Curikin vividly puts it, no computers, no aircrafts, no automobiles, isogenic as they are built, acquire spontaneously personality of their own and respond in a probabilistic manner to environmental cues by all or none functions and or structural transitions. Because it conflicts with the deterministic assumptions of MCC, non-genetic heterogeneity was initially viewed with suspicion as key difference as average cell.
Hang on a second. Okay, here we go. As a consequence
of rogue cell behavior resulting from so-called illegitimate transcription. But as instances of heterogeneity became more widely reported, researchers could no longer afford to dismiss them. As a result, an engineering term began to be used to designate this phenomenon in order to render it theoretically compatible with MCC. This is the concept of noise, which has become widely adopted by the community as a whole. It is worth reflecting for a moment on the consequences of the biological appropriation of this concept. You know what this reminds me of? It reminds me of, was it Copernicus or maybe somebody before him who maintained that the orbits of the planets and everything were circular, but there were all sorts of exceptions to that. So they would create these things called epicycles to explain an inadequate model.
I think that was Copernicus. I think that was Copernicus. I don't think it was Galileo.
And so this once again illustrates that in moving from one paradigm to the next, gaining wider acceptance, there's this point in time where you invent exceptions, almost like special pleading, as to why. Well, we won't really count this as pointing to the need for a new model or a new paradigm. We'll just say it's noise, and that way the same principles are true, but occasionally you get noise, and that interferes with things.
Yeah,
right. Yep.
But it's still important in that it sort of brings Shannon information concepts into cell signaling, and that's one of the things. I'm not sure where he's going with this, but let's see if that's it. Continue.
Oh yeah. I'm still it. So I'm on more recently now.
Yeah, I think so.
More recently, however, there has been a noticeable shift in how the biologists speak about noise. discussions about how cells tolerate noise are gradually giving way to discussions about how cells exploit noise. The reason for this is that as research into the non-genetic heterogeneity of cells continues, evidence for the biological importance of this phenomenon is mounting ever rapidly. We now know that non-genetic heterogeneity plays key roles in both microbial and eukaryotic cells in embryonic development and in evolution. For one thing, it is a crucial generator of phenotypic diversity, which enables cell populations to adapt rapidly to changing environmental conditions. It does so by permitting the implementation of probabilistic diversification strategies within a population, such as bet hedging and divisions of labor, which can confer considerable fitness advantages. It also influences cell fate decisions, which facilitates the regulation of differentiation during development. Non-genetic heterogeneity has even been suggested to allow tumors to counteract the effects of chemotherapy, thereby limiting the efficacy of target selective drugs. In general, it is clear that highly heterogeneous cell populations are more robust and they adapt, grow, and evolve faster than more homogeneous cell populations. Far from being a nuisance, noise, it turns out, is central to many cellular functions. One very important, I'm sorry, you want to say something?
No, go ahead. You're good.
Okay. One very important theoretical implication of the probabilistic nature of cellular behavior and the observed heterogeneity of cell populations is that quite literally every cell in an organism and elsewhere is a unique entity. No two cells are identical, given that no two cells respond to a stimulus in the exact same way, even if they are genetically the same. As this simple yet profound observation becomes more widely recognized, cell individuality is likely to become an ever more important area of research. Our traditional dependence on methods that average out responses across populations has inadvertently driven us to rely on what Levsky and Singer fittingly call the average cell, a statistical contrivance for representing biological knowledge beyond the limits of detection. But as these authors point out, the advent of single molecule methods has demonstrated that the average cell is a myth. Variability is everywhere in the cellular world. In fact, as a cellular phenomenon, variability is less difficult to explain than similarity. Looking to the future as cell biology progressively morphs into single cell biology, and we devote increasing attention to carefully characterizing not just individual cells, but also individual molecules in individual cells, we may soon find ourselves in the position of having to reconsider our understanding of even the most basic biological processes. The following excerpt, co-authored by one of the leading figures in the field, effectively summarizes the two very different views of cellular behavior that I have examined. As biologists, we must grapple with and reconcile two very different views of cellular behavior. On the one hand, we frequently think of cellular functions as being determined by circuits of interacting genes and proteins. In a loosely analogous way to electronic circuits, these chemical circuits encode genetic programs that underlie differentiation, the cell cycle, and other behaviors. They accurately respond to stimuli and generate precise behavioral programs in individual cells. On the other hand, there is the noisy view of the cell we get when we actually look at cells. They exist in squishy, dynamic, and heterogeneous populations. The morphologies, gene expression patterns, and differentiated states of which differ from one another even when environment and genotype are fixed. The main differences between these two conceptions of cellular behavior I have discussed in this section are summarized in Table 4.
Okay, so here's Table 4.
Mm-hmm.
Oh, does somebody want to read Table 4, or should we just allow people to look at it? Individually predictable versus collectively predictable.
They can look at it on their own. It's on the screen. If they want to do that.
Okay, so we got the conclusion left, and
All right.
and it's Immutable's turn to read the conclusion for us.
Oh boy. Conclusions towards a new view of the cell. I have argued in this paper that molecular biology is currently undergoing a fundamental shift in its theoretical conceptualization of the cell. The conventional, mechanical, reductionistic, and deterministic view is gradually giving way to an understanding of the cell that emphasizes its fluidity, plasticity, and stochasticity. Faced with the formidable task of interpreting the vast and ever-growing amount of experimental data that continues to get published, explanatory appeals to engineering notions of designs, programs, and circuits are increasingly replaced by recourses to physical principles of non-equilibrium dynamics and complexity theory. Cells are empirically revealing themselves to be inherently dynamic, self-organizing systems that respond stochastically and non-linearly to environmental stimuli. Give me 1 second, I need to charge my computer real quick.
An editing moment. So let's see. Scott, so what we should do is after we stream this, maybe start another live stream afterwards and see if we can get people to come in.
Sure.
Yeah, we only have about, we don't have that many people viewing it. I don't know how to increase viewership or viewer interest other than to maybe show some illustrations eventually. We'll get better at this as we go along.
Yeah, I don't know. I like the pure idea of seeing what the scientists are saying in their own context. And I know we've references to creationists off and on, but mainly this has been more about what's happening in science. What is this one paper, this one author trying to tell us? How interesting is it? And what does it do to increase your knowledge about the subject?
Yep. We ready to continue, Immutable?
Yep.
Okay, go.
All right, 1 sec, full screen. Here we go. The inescapable conclusion that follows from the analysis I presented is that the cell can no longer be unproblematically conceptualized as a machine. Over the course of the paper, it is...
Whoops. Let me... Go ahead. Whoa.
It has become apparent that cells lack all four characteristic properties of machines that are identified in the introduction. First, once their crucial role that self-organization plays in shaping the cellular architecture is acknowledged, it is difficult to uphold the idea that spatiotemporal arrangement of parts of a cell obeys a predetermined blueprint or design, as it does in a machine. Second, the conformational flexibility of most cellular constituents and the functional promiscuity that they exhibit shows that a cell's operation is not as tightly constrained by its structural configuration as it is in a machine. Third, whereas a machine performs its function by precisely following a predefined sequence of steps, a cell can arrive at a particular end in a variety of ways. It can recruit different kinds of molecules to the same function, or the same kind of molecule to different functions, depending on the conditions it finds itself in. And fourth, a cell cannot be broken down into parts without jeopardizing its structural integrity in the way that every machine can. Cellular components form deeply intertwined, ever-changing networks of interactions that cannot be individually dissected without sacrificing the organization of the whole. Cells are not engineered systems of discrete interacting computational components naturally yielding to compositional analysis, which is why they cannot be fully explained reductionistically, and neither do they operate deterministically, which is why their behavior cannot be perfectly predicted.
Cool. Monad was wrong. Go ahead.
Monad was wrong. The cell is not a machine, but something altogether different. Something more interesting, yet also more unruly. It is a bounded, self-maintaining, steady-state organization of interconnected and interdependent processes. In an integrated, dynamically stable, multi-scale system of conjugated fluxes collectively displaced by thermodynamic equilibrium. Given its precarious nature, the cell is constantly having to negotiate a trade-off between structural stability and functional flexibility. Too much rigidity compromises physiological adaptability, and too much promiscuity compromises metabolic efficiency. The cell accomplishes this by continuously turning over and reorganizing its constituents into different macromolecular complexes with diverse functional capabilities, which assemble and disassemble in order to meet the ever-changing demands of the environment. The permanent stochastic shuffling of molecules inside the cell and their opportunistic associations to form transient functional ensembles in response to intracellular and extracellular cues provides fast and robust solutions to the adaptive problems faced by the cell in a way that strikes an optimal balance between efficacy and plasticity. Although this view of the cell has only come to the fore very recently, it is rather surprising to find that the theoretical principles that underlie it, as well as the empirical findings that support it, are not new at all. General denunciations of the MCC go back well over a century, at Haldane 1884, and even the most recent empirical discoveries in each of the four domains I have examined in this paper have unmistakable historical precedents. For instance, in the first half of the 20th century, it was not unusual for biochemists to describe the cell and its obstinately solid and rigid constituents in terms of streams, fluxes, and other processes. A particularly visionary characterization of the dynamicity of the cellular architecture was offered by
Heard of somebody.
somebody.
It'll help if I can. Bertalanffy?
It was offered by Bertalanffy, who is one of the first theoretical biologists, though today he is better known as the founder of general systems theory. Formations such as the nuclear spindle, the Golgi apparatus, and the like appear as structures when we have them before us in a fixed and stained microscopic preparation. However, if we consider them and their changes in time, they are a manifestation of processes at the chemical and colloidal levels, quasi-stationary states that last for a while but soon undergo changes or disappear. Challenges to the undue emphasis on the structure and specificity of proteins are likewise nothing new. The first reports of proteins with disordered structural domains date back to the 1950s. And some hypotheses regarding the substrate ambiguity, and catalytic promiscuity of metabolic enzymes are over 40 years old. Similarly, the suggestion that a microscopic ratchet might be able to harness the energy of Brownian motion to generate directed movement was carefully explored by Feynman in his physics lectures more than half a century ago. Moreover, experimental evidence for the stochastic nature of cellular behavior goes back 6 decades. And the heterogeneity of isogenic cell populations was already noticed in the 1970s. Nevertheless, all these ideas and observations have remained severely neglected for many years. Only in the last 2 decades have they begun to receive widespread attention, mostly because of the adoption of novel experimental methods has served to experimentally substantiate them, making them impossible to ignore. But what is perhaps the most surprising of all is that even though one would be hard-pressed to find a molecular biologist today that would dispute the fact that a cell is an open system far from equilibrium, or that because of its microscopic size, the effects of stochastic fluctuations on its operation cannot be overlooked, many continue cellular and molecular phenomenon in terms of the classical mechanics, equilibrium thermodynamics, and mechanical and electronic engineering. That is to say, in terms of principles and concepts that are fundamentally at odds with the physical nature of the cell, this curious refusal of many researchers to accept or even seriously consider the new view of the cell that is arising is likely to be due to several factors. One might be that the new view is less intuitive than the MCC. The MCC, after all, draws on our everyday familiarity with machines. It is not natural for us to interpret everything it is almost natural for us to interpret everything in mechanical or engineering terms, because such interpretations accord well with our experience of the familiar macroscopic physical world that we and our machines inhabit. Consequently, confronted with a microscopic entity such as a cell, the challenge for researchers is to look beyond our usual engineering principles and to appreciate the less familiar logic of biological organization, uh, obtained from Glick, 2007, page 132. Another factor that may help account for the reluctance of some researchers to endorse the new view is that it appears to make the cell a harder object to study than the MCC. Viewing the cell as a machine allows us to think of its organization in terms of modular, solid-state circuits that can be approached reductionistically, and it also gives us the confidence to expect that when we eventually work out how well all the cell's parts fits together, we will be able to completely predict its behavior. If, on the other hand, we view the cell as highly integrated, self-organizing fluid system comprised of densely interconnected processes, ever subject to stochastic fluctuations, we no longer have reasons to suppose that achieving such epistemic goals is even possible, let alone feasible. The stark contrast between these two outlooks is exemplified by their strikingly different ways of understanding causation in the cell. And it serves to explain why some researchers find it easier than others to obtain funding and publish their work, as Mayer et al. Pointed out. It is much easier to write and publish a paper suggesting that protein X is necessary for transmitting a signal from A to B, than showing that protein X is one of the many potential components of a heterogeneous ensemble of signaling complexes that together couple A to B. Mayer et al. 2009, page 81.6. A further factor could be that accepting the new view of the cell requires us to adopt and maybe also develop concepts that fall outside the remit of conventional molecular biology toolbox. It requires us, among other things, to seriously consider how the ideas of non-equilibrium thermodynamics and complexity theory, and even those of condensed matter physics and quantum mechanics, may be brought to bear on the interpretation and explanation of the phenomenon we investigate. And this might not be agreeable to all researchers, many of whom appear to show little appetite for theoretical considerations, or worse still, assume that they can proceed in the absence of the theory altogether. Despite all of this, the advantages of embracing the new view of the cell are legion. Most importantly, the new view gives us a systematic and internally consistent interpretative framework capable of making theoretical sense of a multitude of empirical findings that appear paradoxical and almost inexplicable when viewed through the traditional lens of the MCC. Reports of self-organizing organelles, liquid-like macromolecular assemblies, fuzzy signaling complexes, moonlighting proteins, non-mechanical motors, order from disorder processes, non-genetic heterogeneity, and cell individuality seem totally baffling from the perspective of the MCC, but they can all be perfectly accommodated within the interpretative framework that is currently emerging. Findings which are confusing and unexpected within the old view become natural and expected within the new one. Ultimately, the current practice of overlooking some of the principles that govern the internal operation of the cell because they are unfamiliar, and of dismissing many of the cell's distinctive properties because they are difficult to study, is likely to be a mistake. Only by confronting these head-on can we hope to one day arrive at a theoretically satisfying understanding of what the cell is and how it functions as an integrated unit.
Okay. We did it. Read a paper.
Holy crap.
Yeah, that was incredible. Let me go and find something here. Did I not find a book by Nicholson? Was that by him, or was it?
I think you said you did. Of course, we have a whole list of his papers in a link somewhere.
Yep, I was going to show that.
Your camera is frozen.
Oh, again.
Don't be a Leo now.
God damn it. I don't know what is wrong with StreamYard that it does that. But the camera is, yeah, I can see that it's not frozen on, it is not frozen on the computer. It's only frozen on StreamYard. So it is a StreamYard.
Leo was right.
StreamYard.
Leo's right. Yeah, Leo's right.
Don't tell him that. Don't let him know we said that.
Okay, there is a book. So, I believe I ordered a copy of it. Let's see here. Chrome tab, Daniel J. Nicholson. Share. Sharing that. Let's see if we can see that. It would be nicer if we could blow it up a little, maybe. Okay. So the book is Everything Flows Towards a Processual Philosophy of Biology. And that might be something worth reviewing. Then he has this gigantic paper, 87 pages on nature and normativity, which Danny got excited about. And then he has, of course, he's the Cell and Machine, and On Being the Right Size Revisited. So he's got a lot of stuff on this.
Well, the problem with engineering metaphors and molecular biology. That's an interesting idea.
This is God, Otangelo must. He's probably. He probably feels like. I mean, even though we're recording this and it's not live yet, he probably feels like something's wrong in the universe. Something is wrong in the universe.
He feels a wiggle in the force.
The force, yes.
The cytoskeleton of the force has been disruptive.
Oh, the machine analogy. Oh no, it's being threatened.
Every biologist thinks that the cell is a machine, except for quite the number of them who don't think that.
I know. Well, it looks like it's been a parallel idea all along, but for just because it's so sexy to call it a machine or a factory or whatever, it's just been ignored.
Yeah. I had this idea that hit me probably 6 or 9 months ago. I noticed that biochemists were more likely to push back or to be ideas, to believe in intelligent design than molecular biologists. And it'd be interesting if somebody actually did a survey and tried to figure that out, because biochemists are used to looking at pathways, right?
Well, yeah, so are molecular biologists, but we're doing it in a different way.
Molecular biologists look more into what is happening chemically and, in populations of molecules rather than looking at those pathways all the time. Biochemists, that's pretty much their job, is to look at the pathways. Whereas molecular biologists kind of have a different slant on it. It's just a wild idea that I had that, that may have some sense to it. Particularly if all you've studied is biochemistry and you've never, gone into looking more deeply into the molecules themselves. I don't know.
Oh yeah, I mean, I've known some biologists that can't draw out molecules very well, and that's like, yeah, that's a little concerning.
Yeah,
being something molecular in nature and you can't draw out the molecules, that's a little disheartening.
Yep. So we did a paper. So I think what we should do is run this last stream, which is going to be an hour and 15 minutes long, and then maybe open up a live broadcast after that. If there are enough people around and we can generate some interest.
The Paper Trails after show.
Paper Trails after show. And we'll invite Otangelo and Sal. We'll invite Otangelo and Sal. Yeah. That would be a shit show to remember.
That'd be nice to get Daniel Nicholson in on that, but like you said, he may be too well respected.
Yeah, well,
most of us.
He is in Eastern Europe, I think, so we may have a language barrier.
Of course, this says George Mason University philosophy faculty member. Up on the top of this.
George Mason University, so that doesn't sound like Eastern. But I thought I read that he was that he resided somewhere in Eastern Europe.
Oh, he could be. Who knows?
Not sure where. I think it actually had. Yes, he's in Austria. Oh. That's his address.
Oh, weird. So I guess he does it Zoomy wise or something.
I don't know.
Or he travels.
He looks really young, actually. Though he looks like what, 30s?
Very young.
He does, but then that could be, older picture. Let's see. I'm trying to see what the dates on his papers are. They don't give them.
There's another. Like if you list them in Google, they'll have the dates. Like if you go to Google Scholar and type in Daniel J Nicholson,
Yep.
it'll give his papers.
Let me not type it in. Let me do the trick.
I'm sorry, I should have said input.
Whatever.
If you input it in.
You gotta use Boomer Talk with me. Okay. Am I on Scholar? Am I on Scholar? Yeah.
Are you on Scholar?
I am.
There's Google Scholar there. User profiles for Daniel J. Nicholson up at the top.
2012. Click that.
Yeah.
How many citations has the paper we just read got? I'm curious about that.
Let's see. Okay, I think this was one of his earlier papers. 2013, 2012. Yeah. And we got 28.
From 2010, and I think one from.
Are they organized by date?
Where is the cell really a machine right here? Cited by 39, and that was 2019, so that's pretty recent. So he seems to have started in about 2012. Is that
or 2010 because there's a 2010 paper there.
Yeah, 2010. Yep. So anyway, he is worth looking at a little more seriously. He's got some pretty good ideas, and looking at his book, Everything Flows. That might be really good. That's available as a PDF. So all of this is open access. There isn't anything here that, except for this one here, neither logical empiricism nor vitalism, but all the rest are open. So interesting. Okay, guys, we can wrap her up.
All right, let's get her done.
All right, Bill.